All right, thank you. I was successfully able to hook up my laptop for this presentation, so this means I am now qualified to receive my PhD. I'm not? No?
You need to mute your...
Okay, I am now able to receive my PhD because I can start giving this presentation. Beautiful. Thank you all so much for being here. My name is Toby. I am a graduate student in Steve's lab, and I'm going to tell you a little story about one of the projects I have worked on. This just recently got accepted into Nature Communications and is also available on the bioRxiv. Thank you. And this is talking about structural investigations for how the potassium channel TWIC1 is able to gate in response to the pH at the selectivity filter. And if you don't know what any one of these terms are, then I am going to go through every single one of them. So for starting off, the system that I work on is on ion channels. And essentially for ion channels, these are molecules that are going to be responsible for conducting electrical charge through a cell membrane. And they are able to do this through allowing the passage of charged atoms known as ions. And this can include calcium, sodium, potassium, and a large number of other ions such as chloride. And essentially the movement of these charged atoms through a cell membrane leads to electrical charges moving through the membrane, allows for electrical signaling. This is responsible for cell signaling in the brain. It's responsible for the movement of our muscles, especially our heart. It's involved in a wide variety of critical cellular processes such as cell division and numerous things with which life can exist. And what I wanted to study are some specific structural aspects of potassium channels. And the thing that got me interested into potassium channels is that whenever you look at the vast majority of potassium channels that exists in nature, there tends to be a very common and very stereotyped structure. You have these 5 amino acids that have very little variance between different species that are able to coordinate these potassium ions that are shown in orange. And essentially, whenever you look at an ion in solution, it's not just a free-floating positive charge, because this electrical charge is unstable and needs to be stabilized. And what's actually stabilizing it are these partial negative charges that are present on an oxygen atom. Some of the lone pairs on this oxygen atom, some of the free electrons will actually point towards the positively charged ions and help stabilize them. And a potassium channel works by essentially doing something very similar. You have this peptide backbone containing all of these carbonyl oxygens shown in red. They point towards the potassium ions and they help stabilize them at a distance that potassium would normally find an oxygen atom in water. So to a potassium ion, this looks very similar to its own hydration shell. But if you take an atom that is smaller than potassium, such as sodium, it actually can't move through the pore very well. This pore is too small for a fully hydrated sodium to move through, but it's also too big for the dehydrated sodium to move through, so it's able to exclude atoms that are not the exact size as a dehydrated potassium. And this structure is not a static structure. It's actually able to move in a variety of systems. So the selectivity filter is known to gate in response to a large number of different stimuli. This has been especially well studied in KV channels, voltage-gated potassium channels. And while this has been functionally shown numerous amount of times, there's very few instances in the literature of a potassium channel C-type gating and having structural knowledge of how it does that. So I wanted to see a potassium channel that gates at the selectivity filter in response to a physiological stimulus. And the specific system I was working on is TWIC1. So this is a member of the 2-pore domain potassium channel family. They're called 2-pore domain because most potassium channel subunits will assemble as tetramers, so 4 subunits per channel. There's going to be 1 pore subunit per channel, and each one of the carbonyls from the subunit helps to coordinate the potassium ions. These are dimers. You've essentially taken one copy of this and you've made another copy, and now you have two pore subunits instead of one. This channel, TWIC1, is incredibly important for regulating the release of insulin from the pancreas, and it's very important for helping to maintain the cardiac rhythm. So it's really important to study for things like diabetes and for heart disease. And one of the things that makes this really interesting is that it actually has a moving piece located right on the selectivity filter. So this histidine has been shown to be functionally important for sensing pH, and TWIC1 is a pH-sensitive channel. If you take this histidine and you mutate it into another amino acid, such as, for instance, an asparagine, then instead of having a channel which will slowly close as the pH drops, you have a channel which is going to remain open all the time. And this is also in a selectivity filter, which is actually one of the few instances in nature of a mutated selectivity filter, where normally this position right here is an isoleucine or a valine. It's a polar threonine here with a hydroxyl group on it. So I wanted to see how this mutated selectivity filter would move in response to pH when the pH sensor is directly located on the selectivity filter. When I embarked on this project, there was already a structure that had been solved at this channel, but I had left open some questions. For one, the channel was characterized at a high pH, but it was actually closed. There was a block that was attributed to either these detergent molecules I'll get into later that helped to purify membrane proteins, or it was blocked by a lipid from the cell membrane. We couldn't tell because of the way this experiment was set up, but we essentially had the protein in a state where it should be open, but it was closed. And what I essentially wanted to ask, since there was no clear connection between this histidine and the block intracellularly, is what is the connection, if there is one, and whenever the pH changes, exactly how is this histidine going to move? Now, in order to answer these questions, there are two key pieces of technology that I utilize. For one, we want to see if this intracellular block is physiological, if this is actually caused by a phospholipid. And in order to increase our chances of seeing something like that, we need to make sure that there is a maximum number of phospholipids that are present. So what I did is I took my protein TWIC1 and I embedded it into something called a nanodisc, which kind of looks like if you were to take a cookie cutter and just chop off a little disc from the cell membrane. That's not actually, sadly, how you're assembling these, but that's what they look like. So essentially, whenever I am purifying TWIC1, I'm starting out by surrounding it by all these molecules called detergent. These are basically similar to what you're going to find in soap, except the big difference between this and soap is incredibly harsh. That would kill all of your protein. This is very gentle. It solubilizes the protein, but it keeps its structure mostly intact, so that way you can study how it works. So I can add in detergent and extract this protein from cells that are expressing it, and then I can start to slowly add in phospholipids that would want to form a bilayer. I then add a scaffolding protein, which is going to surround this disc. The scaffolding protein, by the way, is made from apolipoprotein E, which helps to sequester fatty acids and cholesterol inside of your blood. And approximately 30 years ago, a group of researchers decided, hey, I can probably make discs instead of globules, which will be really useful for biochemistry. I add that in, and then using hydrophobic beads, which work through biochemical black box, black magic, I'm able to specifically sequester these detergent molecules, somewhat specifically sequester these detergent molecules using beads that have affinity for them and have very little affinity for phospholipids. And at the end of the day, I have my protein embedded in a disc with phospholipids and with very few detergents, and if this block occurs, I can be sure that there is a maximum chance that this is due to a phospholipid and a much smaller chance that it's due to detergents. Now, the second piece of technology I used is cryo-electron microscopy. So essentially what I'm doing is I'm taking my protein, I'm putting it in a nanodisc, and then I am going to flash freeze it and look at the flash freeze sample under an electron microscope. Why am I using an electron microscope? Because electrons are really, really small, and so is my protein. If you look at this with a light microscope, then essentially you can't do that because the wavelength of light is a massive number of times larger than what a protein is. Why am I flash freezing this? Well, I'm going to need my molecules to be really cold and really still and not moving around. And in order to do that, I have to freeze it. But if I freeze it very slowly, then I generate ice crystals. That kills my protein. I'm going to take a bath of liquid ethane. I'm going to cool it with liquid nitrogen, so it's around -200 Celsius, and then I'm going to flash freeze it in an instant. So that way, all of the ice doesn't have time to form crystals, and it looks as similar as is currently possible to a native aqueous solution. Once I do that, then I can start looking at my protein. So this is an image directly from one of my micrographs, and this incredibly grainy pixelated image will show you individual molecules of my channel inside of nanodisk. So right over here, you can see a little something sticking out of this disk, and that is my protein embedded within a nanodisk. This is another example where you can see the disk here, and the large extracellular domain of this channel is sticking up out of this side. And the way that I am going to make a structure out of these individual images is first, I'm going to pick individual particles that look like a channel in a nanodisk. You really start out by doing this manually for 1,000 particles, and then you use neural nets to help it fill in the rest, because you're going to end at the end of the day, at this very first step, get millions of particles. You're not going to do all that manually. So we use that part for a neural net, and then afterwards, we're going to classify all these individual images to give us views that have a similar tilt and a similar orientation. We're going to start in 2 dimensions because this is much quicker, but then at the end of the day, we're going to do the same classification in 3 dimensions, where I can separate out, yes.
Your slides are not progressing on the Zoom link. I'm so sorry to play.
Oh, wow, that is surprising and also very annoying. Thank you for bringing that up. Okay.
Since we're recording, I figured you might want the, so if you can come see what type of view it is. Oh, there we go.
Actually, now?
Yeah, it's showing your view, but it's working better.
Okay, cool. This will work then. Okay, so essentially what you're going to do is you are going to, not yet, what you are going to do is separate out all these 3-dimensional views that have your channel from all of the 3-dimensional views that don't. And essentially, I like to think of this in terms of like you're taking a bunch of pictures and you're making a sculpture from it. So the way that I like to think of cryo-EM is imagine that you're like you're a fashion model doing a catwalk on the runway, and people are taking pictures from you at every single angle, and you take all those pictures and you make a 3-dimensional sculpture. I include this skip specifically because, well, that was just fun, and also sometimes what you're going to do is not just see a 3-dimensional sculpture of the protein. You don't want a protein that's static because proteins aren't static. You want to see them moving. So in this instance, what I really want to see is my protein in 2 different conformations. Like if I want to know how this model is doing this really cool dance, I want to see this model whenever she's like this and whenever she's like this, and how she's actually able to move her feet like that. So in the case of trick 1, I want to see this channel when it's open and when it's closed, and then think about that movement, because that is the key for how we can actually determine a biological mechanism for a lot of these channels, is thinking about how they move. So essentially using this process, I was able to get the structure of this protein embedded within nanodiscs to around 3.4 angstroms resolution. In all honesty, there's a little bit of a difference in the resolution because of some other technical concerns I'm not going to get into right now for time limitations, but you can clearly see some differences at the selectivity filter whenever the pH changes. So at a high pH, you're going to see all of these ions that are in gray passing the full length of the selectivity filter. This mesh that I'm showing you is the data that I got from cryo-EM, and then the blue and the magenta models are the models that I built into this data. And you can see that at a high pH, the selectivity filter is completely clear, and at a low pH, it is blocked shut. And whenever you look at this model from the top down, extracellularly, you can also see that there is a very clear protein block of the channel, which is caused by this threonine that's sticking right up above the pore, and is stabilized by a histidine that's interacting with this aspartic acid. This aspartic acid, by the way, is very highly conserved. It's present in every single 2-pore domain potassium channel, and present in most of the other subfamilies of potassium channels. And you can also see an actual movement of this protein whenever you just make a morph between these two states, where this histidine comes up and forms the salt bridge, and then this threonine right in the middle is going to stick right above the pore and prevent any ion production. Now, one of the things that surprised us whenever we first got this data is this movement propagates throughout the entire rest of the channel. It's not just the selectivity filter that's moving, but the entire channel gets stretched out perpendicularly to the cell membrane as if you took two tweezers on it and started pulling on it. And the reason this movement happens is because of the positioning of this extracellular domain above the selectivity filter. So if you look at the position of this histidine at the high pH state and the low pH state, it's moving upwards by around 6 angstroms, and the extracellular cap is only 4 angstroms above the selectivity filter. So if this cap doesn't move, then you're going to get a clash between this histidine and the extracellular cap. In order for the cap to move, it needs to start moving all the transmembrane helices because the cap is directly connected to the transmembrane helices. In fact, the first transmembrane helix is part of the same kinked helix that forms the cap. So as this histidine moves upward, the cap moves upward, all of the rest of the transmembrane helices have to move in response to that, and this is connecting to the intracellular lipid block. So essentially, this intracellular lipid is accessing underneath the pore through a lateral membrane opening that connects to the intracellular leaflet of the phospholipid bilayer. And this is something that's present in all K2Ps, and for most of the other K2Ps that we've characterized, at least for the 2 instances where we do have structures of the K2P in the open and closed conformations, this lateral membrane opening is completely shut whenever the channel is open, and there is no instance of the lipid being able to access it. And then whenever you have the channel in a closed state, this promotes the opening of the lateral membrane cavity, so that way this lipid can come underneath and block the channel. For TWIC1, this is still mostly the case, except that at a high pH, it's there. The lateral membrane opening is still kind of there. It's right at the cusp of what you would need in order to stick a lipid in there. It would only require a subtle rearrangement, and this is likely the reason why a lipid block was observed in this previous structure. So the lipid is connected to the histidine through this propagated movement, but it is also something that can still happen even at a high pH. And that is the conclusion of this talk. So essentially, the acidic pH is going to promote a block of the selectivity filter through this histidine, but the histidine is also connected through a previously observed lipid block because the entire structure has to stretch and dilate as this histidine moves. And with that, I will get into what is also my favorite part of these talks. So a huge thank you to all the members of my lab, and especially to Steve for being such a wonderful mentor and allowing me to have the freedom to go in so many different directions as a scientist while I was working here. Thank you to everyone else in the lab for all of your collaborations, for all of your discussions, and learning how to do good science. Huge thank you to everyone at the Cal Cryo team for their assistance in data collection. Huge thank you to this person who was my undergrad RA, Vivian. She's currently in her 3rd year of medical school right now. Also, huge thank you to this person. This is my dad, Carl. He passed away in December 2019, and throughout his entire life was an example to me of resiliency and persistence in the face of adversity. Additionally, just last year, my uncle Steve Reynolds sadly passed away from the COVID-19 pandemic. I do not have any pictures of him on my phone. They were both huge inspirations to me growing up and throughout my time in graduate school. Another huge person that was an immense source of support for me is my brother. That's him, Trevor. That's my 2-year-old niece, Leah. That is Thunder. He just turned 10 yesterday. They have been a constant support to me. He is one of my oldest friends in the entire world. And also a huge shout out to many of my other friends who are here today. Thank you so much. You have given me a sense of community I have not felt anywhere else, and have supported me in numerous ways throughout my life in Berkeley. And with that, I will take any questions. Thank you all so much.
Might be able to squeeze in one question. We're starting to get over time, but. Any questions? Anyone from the online audience have a question? Not seeing anyone, so next up is Amanda.